An American biochemist was awarded the Nobel Prize in Chemistry in 1993 for his investigation of the polymerase chain reaction (PCR). This groundbreaking technique revolutionized molecular biology and genetics, leading to the significant advantage in genetic research, forensic analysis, medical diagnosis. His work demonstrates the power of investigation thinking and interdisciplinary collaboration in a driving scientific process. Kary Mullis invented the polymerase chain reaction (PCR) technique in early 1980s. This technique involves denaturation, annealing, and extension, allowing researchers to amplify DNA sequencing in a test tube. PCR has revolutionized genetic research, medical diagnosis, forensic analysis, and biotechnology by enabling the study of change. Genetic diseases involuntary relationship and pathogen detection. Mullis' creation exemplify the power of involving thinking and interdisciplinary collaboration in a scientific discovery, shape the way scientists study and understand the genetic makeup of organisms. His groundbreaking work has left an indelible mark on genetic and molecular biology.
In 1976, the Yanqian, a Chinese scientist, discovered stable DNA polymerase, which made a fundamental contribution to the development of PCR technology. This chapter mainly discusses about the principal characteristic, common problem, and their solution of PCR. The different types of PCR techniques including traditional PCR, quantitative PCR, qPCR, digital PCR, dPCR, and their application for rapid detection.
Yeah, I think it was with a bag similar to this.
PCR is critical technique in forensic DNA analysis which enables the amplification of small amounts of DNA to create enough material for analysis. For example, PCR is used in short tandem repeat STR analysis, mitochondrial DNA mtDNA analysis, and single nucleotide polymorphisms SNP typing. For further information, PCR used to amplify variable number tandem repeats VNTR and short tandem repeats STR using primers that flank each of the repetitive regions. Instead, a set of alleles for each person will be found by analyzing the size of the fragments obtained from any individual for each of the STR. Additionally, the PCR-based genetic or DNA fingerprinting methods has been powerful in identifying criminal suspects due to analysis of evidence at crime scene when samples of DNA isolated from a crime scene and compared to suspects. Through its specialness in using only small amounts of sample, it is often applied when only a trace amount of DNA available, such as for analysis of ancient DNA. The success of this technique is shown in archaeology fields such as identification of a Russian Tsar, the body of English King Richard III, and Egyptian mummies, also used on animals such as 40,000-year-old mammoth. Furthermore, quantitative PCR often applied to determine levels of gene expression quantitatively as it enables the estimation of the amount of a given sequence present in a sample. qPCR also allows the quantification and detection of a specific DNA sequence where consists two methods. More interesting fact, they have combination technique called RT-qPCR, which is combined reverse transcription and qPCR. In this term, mRNA is converted to cDNA and then quantified using qPCR. This technique used in laboratories follows the possibility of error at the end point of PCR and increasing chances for detection of genes associated with genetic disease. These foundations will found accurate fitting methods of experimental data in research, such as in analysis of wireless fidelity radiofrequency radiation on the expression of E. coli genes, and analysis of gene expression as a potential biomarker for early-stage diagnosis in colorectal tumor and cancer patients. Applications of PCR also can be seen in detection of infectious disease. For example, PCR can detect viral DNA because of the high sensitivity of PCR permits virus detection before and after infection disease. Through this, the amount of virus in a patient can be quantified by PCR-based DNA quantitation techniques, which is a variant of PCR-RT-PCR used for detecting viral RNA. In this step, the enzyme reverse transcript test used to generate a DNA sequence that which match the viral RNA. Then DNA amplified as per the usual PCR method. This method widely used to detect the SARS-CoV-2 viral genome.
And now it's pulling a molecule out of this bag.
First advantage of PCR is high sensitivity. PCR can detect even minute amounts of DNA, making it highly sensitive for various applications, including early diagnosis of diseases and forensic investigations. Is speed. PCR can amplify DNA in a matter of hours, which is significantly faster than traditional cloning and amplification methods. Effectiveness. Once established, PCR is relatively inexpensive compared to other molecular techniques, making it accessible for many laboratories.
But your body knows it extremely well.
First is contamination risk. PCR is highly susceptible to contamination, which can lead to false positive results. This requires rigorous laboratory practice to prevent cross-contamination. Second is temperature control. PCR relies on precise temperature cycling, requiring sophisticated and well-maintained maintenance. Thermal cycle, which can be a limitation in resource-poor settings.
Pig heart valves have lots of these on it. Is the reason you can't.
Firstly, polymerase chain reaction PCR amplification of a nucleotide protein gene sequence of infectious hematopoietic necrosis virus. The purpose of this study clearly to use PCR to amplify the target sequence for an oligonucleotide probe that could be used for rapid detection and identification of IHNV. So, they choose a primer pair that flanked the probe binding site by about 100 nucleotides on either side. 7 isolates of IHNV were used, include an isolate from each of the different electrophoretypes. To produce a PCR product of a size that could be easily identified on an agarose gel, 2 DNA oligonucleotide primer were designed using a published sequence of the ENT gene of the round butte strain. In result, obtained that a single band of DNA of the expected molecular weight was observed after agarose gel electrophoresis and ethidium bromide staining after 25 cycles of PCR amplification. Meanwhile, no PCR product was detected, but the PCR efficiently amplified IHNV RNA at all starting template concentration test within 13 or more cycles. In addition, the PCR amplification of a portion of the IHNV nucleoprotein gene get a large amount of product of predicted molecular weight. For instance, this method will be faster, less costly, and will be valuable for detection of latent or integrated viral pathogen. Because of that, the combination of PCR and nucleic acid probe method will have increasing use in fish disease diagnostic. Other than that, the technology represents the use of PCR will increase both application in many aspects of fishery science, such as research on stock identification, gene expression, taxonomy, and forensic.
Secondly, a typical case report droplet digital PCR aids in the diagnosis of children with fever of unknown origin. This study aims to investigate the effectiveness of droplet digital polymerase chain reaction known as ddPCR in identifying pathogens in children with FUO as a novel application. To make this study success, 5 ml of the 7-month-old child's peripheral blood were taken immediately on the day of admission because of FUO. At the same day also, peripheral blood ddPCR analysis, synchronized blood culture, and MNGS analysis were conducted. In result, ddPCR still had a sensitivity of 55.56% and a specificity of 78.75% in patients who had previously received antimicrobial therapy. A study on bloodstream infections has shown the detection rate of ddPCR within the detection range of ddPCR targets is significantly higher than other nucleic acid detection methods such as qRT-PCR and MNGS. Besides, ddPCR is also the detection of polymicrobial infections. For instance, ddPCR could be a promising complementary tool to improve the diagnosis and guidance of treatment in children, such as can help to identify pathogens in peripheral blood samples of patients with local infections, especially of children with FUO. Early pathogen screening and with the ddPCR assay could also be expected to improve prognosis in febrile patients with an unknown cause.
The conclusion of our presentation is that the invention of the polymerase chain reaction PCR by Kary Mullis, who was awarded the Nobel Prize in Chemistry in 1993, has had a profound impact on various fields in science and technology. PCR enables the efficient application of DNA segment from small sample, which is crucial for tools of experimental method in research, medical diagnosis, and forensic science. By facilitating detailing DNA analysis, PCR significantly contribute to advance in molecular biology and our understanding of genetics.